Okay, I think that's our chime. So welcome everyone. Our first talk today is by Jarrod Sopniewski, who's a PhD candidate at the University of Western Australia. Jarrod's work focuses on devising novel methods to understand patterns of diversity. And today we're going to be learning about why estimates of heterozygosity from SNPs are context dependent and often wrong.
Good morning, everyone. Thank you for coming. Thank you also for organising this symposium. I'm excited to get to be the first one to talk today. My name's Jarrod. I'm from Perth in Western Australia, which is about as far away as you can get from here, so sorry if I ramble on a little bit. But yeah, I'm going to be talking a little bit about some of the work I've been doing in my PhD, and specifically about why estimates of heterozygosity from SNPs might not be correct. Sorry. So first of all, why do we bother doing population genomics for conservation? Well, briefly, I guess if we think about a study area, a piece of the landscape, and a species that occurs across that study area, if we can get DNA from that species, we can tell a lot about that species. A lot of the time these days, for conservation genetics, we're going to be getting DNA in the form of single nucleotide polymorphisms, or SNPs. So just a brief reminder about what SNPs are. Basically, they're those parts of the genome which vary just at a single nucleotide. And those variations could be neutral or they could be related to variation, so for instance, color differences or size changes. They're basically a really useful form of data which we can use to tell a lot about a species. Once we have that data, we might be interested in trying to work out some population structure, so working where within that species individuals are sharing genes and where there might be barriers to those genes being passed as well. And we can split those into different populations. And once we've split something into populations, we're interested in the difference between those populations. One of the first things we might do is look at genetic diversity, and often we're measuring that in heterozygosity. So when we're measuring heterozygosity from SNP data, essentially we're lining up every sequence from every individual. We're reducing that to the parts of the sequences where there is variation, and essentially we're just measuring what proportion of variation there is within that species, within that population, sorry. So for every population we're interested in, we get a number, and the higher that number is, the more diverse that population is. Easy. The problem is, if for some reason we don't capture all of those individuals we talked about before, or we're just looking at a different set of data, that fundamentally changes how we calculate that statistic. So basically, when we have removed some individuals from the population, we might not have a site that has any variation now. So traditionally that site just gets removed from our calculations, which affects what we're calculating, which of course ends up in a different number. And importantly, the way that number changes isn't always the same between populations. So what that means is that when we're comparing two different populations, they might not be comparable to each other. Those estimates might not be comparable to each other because of different sample sizes. And also within the same population, just depending on what we find, the result we get might not be comparable to each other. So it's leaving a lot to chance, and there's a lot of problems with that sort of traditional method of calculating. Now, I'm not the person that first brought that to attention. Hopefully you've all read this paper, and if you haven't, please go do. But it's a really good paper that really breaks down this problem and comes up with a solution to it. And that solution is calculating autosomal heterozygosity. So briefly, the basic difference between that traditional SNP-based measure and autosomal heterozygosity is that rather than considering just where there is variation throughout the genome, we just consider all the sites. So that means when we are removing individuals, we're not affecting those numbers as much as we might otherwise be. Obviously, that's summarising a really good paper into one sentence, so go read that paper. And it's not my work, so I don't want to go into it too much. But essentially, that paper comes up with 3 recommendations which should result in better estimates, which we need if we're doing conservation genetics across lots of species. And those recommendations are to use that autosomal measure, also to omit sites with any missing data. The reason for that being that it impacts how observed and expected heterozygosity are calculated relative to each other, and also to analyse populations independently of each other. So none of that's new, none of that is my work. But in completely unrelated work I was doing on this nice little Australian flog, which, just as a side, has really fun field work. It just involves driving through the Australian bush and looking in toilets for frogs. Using this method, we had a problem. So we got a measure of diversity, which seemed good, but as we increased the amount of individuals we're checking, we kept getting this problem. Our diversity kept going down. Our results weren't making sense. It was quite perplexing. So we looked at this data a little bit more closely and tried to work out why this might be happening. So with this specific frog, we noticed that its population was especially diverse. So these are quite low values compared to SNP-based heterozygosity, but for an autosomal measure of heterozygosity, these values were relatively high. We also noticed that there was a lot of data missing. So when we are omitting sites with missing data, we're dramatically reducing the data set that we're looking at. And we also noticed that there were a lot of sites which had 3 or 4 variants. So often when we think of SNPs, we just think of biallelic sites. There's a major frequency allele and an alternate allele, but there are 4 nucleotides, so there can be up to 4 variations at one site. And we were noticing that within this particular population, there are a lot of these sites. Now that causes problems because usually when we calculate population genetic statistics using programs like XACS, these 3 and 4 allele sites, they're just taken out of the equation, which is removing some variation. Basically, because we have a lot of missing data, we're removing a lot of sites following this method as well. And we also noticed that because this population was especially diverse, these problems were magnified. We found that where there was missing data, the missing data tended to be more common at sites where there were variation as opposed to fixed sites. So that means as we are getting more and more missing data, basically we're just removing more variable sites than fixed sites, which is causing us problems. This problem scales with diversity, basically. So for 3 populations of this frog, we found that the more diverse in reality that frog actually was, the greater these impacts were. So the reductions were bigger. We also saw this in 2 other species, which I didn't put it on the same scale, sorry, but essentially, as well as between populations, we also saw that across species, more diversity equaled a bigger problem. So how can we fix that? A really simple solution that we came in is just to look at things in smaller chunks. So if rather than considering 10 individuals, for instance, if we just consider 5 individuals, all of these issues that are causing this reduction in heterozygosity are basically just minimized. So as we consider a smaller subset, the chance of a missing site occurring at a site is less because you're looking at less individuals, so you're removing less sites because of that. The chance of there being a 3rd variant or a 4th variant at a site are also less, so you're removing less sites because of that, and we're keeping that population data. And basically that means that we're just minimizing these issues. So it's a pretty simple solution, but it means that we can get a better estimate which is less affected by all of these issues. So basically the central method we're proposing is rather than looking at all the samples you catch when you're out in the field, just break those up into smaller chunks and sort of aggregate your results to get population mean estimates. So in conclusion, we wrote a paper about this, which you can go check out now. And we sort of agree with everything that is mentioned in this first paper. So you should calculate autosomal heterozygosity estimates, just leave SNP stuff behind, it's too problematic. You should omit sites with missing data. The reasons I haven't talked about within this talk, but you just should do that and read that paper to work out why. And you should analyse populations independently. But what we propose in addition to that is just this method of looking at things in smaller chunks and sort of aggregating those results, averaging those results to get sort of population level estimates. Further to that, that problem that we had with the sort of tri and tetraallelic sites, we don't have a solution for that yet. So it's beyond the scope of this study to sort of build a new way to calculate population genetic statistics. Like I said, this work was kind of accidental in a way. So all the things that we're using to calculate population genetic statistics at the moment, just by default, those sort of tri and tetraallelic sites are removed. So what we're proposing is a little bit of a band-aid for that problem. Basically, just by looking at smaller sample sizes, we're reducing the chance that that 3rd or 4th allele pops up in our data set. But that is still there. It's still real variation. And in our paper, we show that the amount of problem that's causing is small, but it's not insignificant. So we might need to, in further work, look at how we're actually calculating these statistics in the first place, and work out whether we should be omitting these sites or not. That's it for me. Please read my paper. And yeah, thank you for coming.
That was a really interesting talk. I'm wondering, for the tri and tetra alleles, those extra alleles, how do you incorporate minor allele frequencies filters in those to make sure that you're still considering those alleles? Because they're probably going to be very, very rare if they pop up.
Yeah. So I guess a fundamental difference between the sort of autosomal and SNP heterozygosity calculations is that I guess how minor allele frequencies work for SNP sites is, so normally in our SNP calculations we're reducing our data set to sites where there's variation observed. So for that autosomal calculation, that minor allele frequency in that sense is not considered at all, because we're also considering sites with no frequency at all. I guess the other way that you can look at that minor allele frequency is to maybe remove sites that might be genotyped incorrectly, I guess. And that's something that we haven't looked at heaps in this paper, just sort of beyond the scope, which is why this solution's a little bit of a band-aid at the moment, but yeah, we sort of advocate in this paper that it sort of warrants more research, I guess. I hope that answers, sorry.
Great talk. I've published a bit as well in this area, and one of the things I was noticing recently is in GATK, there's a way, or there's something around normalizing or something where it can split a tetraallelic or a triallelic site into multiple different SNPs next to each other, which is kind of it creates sort of fake SNPs around it. But is that something that you've looked in it as a possibility as well, in terms of having to keep those in a larger analysis?
Short answer is no. Yeah, I haven't looked into that yet, so, but I'll check it out. I think I'm off the hook. Okay. Thank you, everybody.
Okay, so next up we have Kristy Mualim, who is a PhD student at Stanford. Kristy couldn't join us today, so she will be giving a virtual presentation. Her advisor, Moy, was planning to come and answer questions, so hopefully he will be here and can field those questions. If not, we'll just have a little bit extra time at the end, and I'll try to get the slide changer up and running again. So Kristy's works to develop frameworks to model genetic diversity changes in non-equilibrium scenarios. And today she'll be talking about genetic diversity loss that continues long after habitat destruction halts. This video, the sound is maxed, but it is fairly quiet, so hopefully everyone is able to hear. If you can't, the sound is loudest over here, so you could maybe move to the front. Apologies.
where now the x-axis is time in generations and the colors represent different percentages of habitat loss, we see that the rate of genetic diversity change is startling when comparing 30, 50, and 90% habitat loss scenarios. One way this tracking could be useful is if we have a sense of the percent of range loss for individual species and a rough sense of how much time has passed since habitat destruction. We could then attempt to get an estimate of genetic diversity still remaining within populations, which could help inform targeted conservation strategies. While genetic diversity trajectories are likely going to change across taxa, one resounding conclusion from our work is that even if current conservation practices succeed in stopping habitat destruction, most organisms will continue losing dramatic amounts of genetic diversity long in the future. Knowing this information can help us better plan for habitat conservation programs, help us develop better indicators to assess extinction risk, and perhaps this information could also be used to help inform global biodiversity policy. One clear way could be integrating this framework with existing indicators like the Living Planet Index, or LPI, and the IUCN Red List. For the interest of time, we'll just be focusing on using the Living Planet Index population size projections. First, if we utilize population size decreases as the proxy for habitat loss, we can then make estimates of genetic diversity loss. And for simplicity, we do this in just 2 time points, short and long term. In the short term, we find that the distribution of genetic diversity loss across 5,000 species is bimodal. There's a group of populations at the far right that have lost close to 90 to 100% of genetic diversity. On the other hand, there's a slightly larger proportion of populations that have lost 0 to 30% of genetic diversity. In the long term, this distribution of genetic diversity loss is much more continuous. There is, however, a larger proportion of populations that are projected to lose 80 to 100% of their genetic diversity. To frame these distributions in the context of defining targets of genetic diversity protection, we will use the previously proposed but now removed UN target of 90%. We find that 50% of species tracked in the Living Planet Index have genetic diversity above this 90% target in the short term, but these projections become more dire in the long term, where we find that only 800 species tracked in the Living Planet Index have genetic diversity above 90%. If we were to quantify the amount of genetic diversity lost across all species tracked, this means we have a 12% short-term and 37% long-term genetic diversity loss. And this is well past the previously proposed, but now removed, UN 90% genetic diversity target. But information is power, and knowing how much we've already lost can help us make plans for the future to conserve the amazing biodiversity that exists around us, and perhaps help us figure out what a new revised target for maintaining genetic diversity could be. And with that, I'd like to conclude with our findings. Our work found that genetic diversity decreases long after habitat destruction halts. This is likely going to look different across different taxa and may be particularly concerning for species with quicker generation times, like insects, and less so for species with longer generation times. While I didn't get a chance to talk to you about fragmentation today in this presentation, we also found some cases where genetic diversity may not follow population degrowths. And if any of these themes are of interest to you, please reach out. And with that, thank you all for listening. Thanks my lab for the support and community, as well as my 5 collaborators that helped me think through a lot of nuances in this project. My email is down at the bottom of the slides, so if any of you have any questions, feel free to email me. And with that, one very last thing, my lab mates are also giving presentations in the upcoming days. So if any of these topics are of interest to you, please go listen to their talks. And thank you again.
All right, everyone. We're ready for our next talk. Today we're going to be having Dr. Barbara Mable, who's a professor of evolutionary genetics at the University of Glasgow, who works on the conservation genetics of large mammals and the evolutionary genetics of plants, microbes, vectors, and parasites. Today, Dr. Mable will be talking about inbreeding and conservation. You can probably just get started.
Thanks, Rich. Well, I'd like to start by thanking the organizers for including me in this really interesting-looking symposium today. The work I'm going to talk about today is the work of two PhD students who've recently finished in my lab, Ronald Melia from Tanzania and Deborah Davey, and they had support on whole genome sequence analysis from Anubab Khan, who is a postdoc. So when we think about conservation, in genetics, we often think variation is better. And so there's lots of good reasons why this might be. So preserving genetic variation, we worry in small populations if they lose it, then they might lose adaptive potential. This could be due to mutations that have overdominance. There could be genes where diversity is advantageous, like immune genes or phenology genes. And there could also be inbreeding depression that's caused by inbreeding. And so this can be caused by various things, but including exposure of deleterious recessive mutations, homozygosity for severe mutations. But the problem we know now, what I want to talk about today is the impacts that genomics are having on our concepts about this. So we know that small, isolated, and inbred populations can purge the worst mutations over time. Heterozygosity hides deleterious mutations, so conversely, really diverse populations actually might have a higher genetic load. There can also be local adaptations, so if you mix populations, maybe that will make them less fit if they're combined. And so genetic rescue does increase genetic diversity, but I think we also have to think about potential risks associated with those kind of management practices. So today I'm going to talk about two case studies, Eastern black rhinoceros in Tanzania, and this is the work of Ronald, and Exmoor ponies in Scotland, and this is the work of Debbie with support from Anubab. So in Tanzania, Eastern black rhinoceros, there's only a few remaining populations. So there's some populations they were down to 3 native populations. One of them is a transborder population with Kenya, and then 2 that are in the Serengeti and Ngorongoro conservation area. One of the things about the way rhinos are managed in this area is because poaching is their most serious risk, they're managed in these intensive production zones. So if the individuals try to move to a different subpopulation, they chase them back. And so what this means is there's not much movement between these isolated populations. There have been some rare natural dispersal events, and they've done some translocations. But the translocations, the interesting thing about these is the animals have come from European zoos or South Africa, but they were all originally collected in East Africa. And so they're restoring genetic diversity potentially by bringing back these animals. There's also hybridization with the native populations once the translocations have happened. So what Ronald wanted to know, there hadn't been any consideration of genetics at all in Tanzania, and he wanted to know what the implications were of these past management practices. So just looking at mitochondrial DNA to start with, what you can see here, this is a map of the populations. The colors are haplotypes. And you can see there's not much, even close populations don't really share the same haplotypes. So we know this IPZ strategy has been quite successful at keeping these populations isolated. We can also look at there's only 6 haplotypes that currently are found in Tanzania. 3 of them are native, and 3 of them came from the translocated individuals. So there has been restoration of some of the past diversity in these populations, but because they're not mixing, then we're not really taking advantage of the translocations if the animals are kept in these small, isolated populations. The other thing we know about the consequences of this, so this is a population in Moru. This is a native population. It was down to 3 individuals when they started monitoring. There was 1 male, and he had moved from Ngorongoro, and he mated with 2 females. And what you can see is they're very highly inbred. He's bred with his daughters, his granddaughters, his great-granddaughters, and so this is really extreme inbreeding in this population. And so the mean kinship is about 0.24, so we know that there's been a lot of inbreeding. So what we wanted to do next was to take a whole genome sequencing approach And so Anya Bob helped Ronald with this to analyze the data. And what we're looking at here, so this figure shows we have different kinds of populations. So these are two different native populations, the Ngorongoro population in the south and the transborder population with Kenya in the north. There's translocated populations, and these are we're calling assisted dispersal when we've had translocated individuals that have hybridized with native individuals. And what you can see, as we expect for genetic rescue, there's reduced inbreeding in the translocated individuals compared to the native populations. But what was really interesting about this was analysis of cohorts. So what we're looking at are cohorts of individuals whose parents had different ancestries. And so if we look within the Moro population, this male who had translocated natively, the first generation of his offspring showed a big reduction in inbreeding. So even though these were adjacent populations, the native dispersal actually had about as much impact as the translocations, which is really encouraging if we were to change the management structure. But these are individuals that, so the light purple ones, though, this is several generations later. So this is 3 generations later, and we've lost any potential benefits of the reduction in inbreeding in these populations. The other thing we can look at is we look at genetic load. So what this figure shows, so we're looking at an RXY analysis. So we're looking at the relative load of the first compared to the second in the comparison. And so if it's below 0, that means there's lower genetic load. And so I'm going to focus just on loss of function mutations, which are in black. And what you can see here is that what we're doing in each case, we're comparing this, what we're calling old natural dispersal. So this is several generations after the dispersal event to the others. And what you can see is there's consistently lower genetic load in this population. So remember, this is really highly inbred. It's been isolated for a long time, but compared to even its direct siblings from before, just after the natural movement, there's lower load. There's lower load compared to the population where their father came from. There's lower load compared to the other native population, to the hybrids and the translocated individuals. And so the other thing we know, if we look at the fitness, though, this Moro population that's really highly inbred also has higher fitness than the other populations. So if we look at this population, what we're looking at is age of first reproduction in this case, and compared to the population that their father came from and to the translocated individuals, they actually have higher fitness. So this kind of raises some discussion points about management. So poaching is the biggest threat that rhinos have, and so this management structure so far has been designed for that, but what are the costs of this? So translocations are expensive, not only financially, but also in terms of animal welfare. And so a better strategy could be changing the management practice to open up corridors of dispersal rather than keeping them isolated. The second example is quite a different kind of species. So this is semi-wild Exmoor ponies in Scotland. And so here the Exmoor ponies have been promoted as ecosystem engineers for rewilding projects because they're very resilient. They're very hardy. You can keep them outside. They give birth by themselves without assistance. And there's Debbie's initial work suggested that even in 1 year, you could have an impact on vegetation if you introduce the ponies. So what she wanted to know was, can genetics inform management of this group? And so again, we did the same kind of analysis. So we're looking at whole genome sequences and runs of homozygosity as an indication of inbreeding. And what you can see is the Exmoors are these green ones here, and they're very highly inbred compared to lots of other breeds of ponies and horses in general, and there's variation across the herds. So potentially, because there's variation across the herds, we might be able to mix them in different ways. But if we look at the deleterious mutation load and we relate it to the runs of homozygosity, we can see that there's a negative correlation with deleterious mutation load and inbreeding. So again, there could be some purging of the mutations. They're colored by mitochondrial lineages, and you can see they're kind of spread across, and we're considering loss of function mutations again. Again, then, if we look at the fitness, though, what we can see is that there's a non-significant trend. There's not many individuals where the whole analysis was based on individuals sampled from across their whole range where their Exmoors are kept. But these are based only on one location in Scotland where there's a herd of about 45 individuals, and so these are the individuals where there is both genomic data and individual fitness data. And in this case, we're looking at parasite loads on the left-hand figure, and you can see it's a non-significant trend. There's not many individuals, but we see an increase in parasite load with inbreeding, which might be a negative sign, and we see a decrease in body condition score with inbreeding. But if you look at these points here, one of the individuals that was most highly inbred didn't have any parasites across several years. It also has kind of an intermediate score. You can also see there's a lot of individuals that have no parasite loads at all. And one of the things about Exmoors is, compared to other horses, they have really low nematode loads. So almost all livestock have huge numbers of nematodes that are endemic, but the ponies don't. And so the thing about this breed is that they've been bred for resilience. They seem to have higher resistance to nematodes, and their condition scores, even though there's this decreasing trend, they also tend to have higher body condition scores, especially considering they're left outside all winter. They don't have supplemented feeding, and so this raises some paradoxes. So we always think about reducing inbreeding, but in this case, the traits for resilience and hardiness might be easier to select for because they've been so inbred. So it may be that we're preserving traits that are important. The other thing we know is that, so we've demonstrated again from the genomic data, there could be purging of fitness in these inbred lines. There could be increasing genetic variation, could actually disrupt these traits. And so maybe we don't want to increase genetic diversity if we're going to release all these animals for rewilding projects. Maybe we want to keep the traits that have been selected to make them useful for this purpose. And if we have local adaptation, different herds, so Debbie it's her own herd she was working on, and she keeps hers completely outside in this remote peninsula in Scotland. Other people have them more like pets in corrals, and so they're going to have different consequences for fitness if you combine them. And so there's a trade-off between retention of these potentially ancestral traits they have and adaptive potential to new conditions. So what I was hoping to demonstrate with these, just a very superficial view of these, is just that maybe we need to rethink. We're thinking in this symposium about multiple species, and not every species is the same. And so we probably can't have just one rule for genetic rescue that's going to apply. So if we're going to consider increasing genetic variation, we should consider the history of inbreeding in the populations. And what's really critical and hard to get are actual measures of phenotypic fitness that you can relate. So we have really rough measures so far, but what we really need in the field is to now take our proxies for fitness that are coming from the whole genome data and actually go in the field and test our predictions. And so the next step is really, we're making a set of predictions on what would be the best management, but we want to make sure about what might happen in the future. So I'd like to end by thanking my co-authors, so Ronald and Debbie. Iron Brew is the Scottish national drink. And Anubab Khan, who helped them with their bioinformatics. Umar Rajas helped us with server access. The co-supervisors, Grant and Jason. Elizabeth Kilbride did a lot of the molecular work, and then some undergraduates who helped. And funding, but especially the Exmoor Pony Society, who funded Debbie. The Zurich Zoo, who funded Ronald. And Leverhulme Trust, who funded Anubab. Thank you. Yeah. Yeah, so this is what I showed you, because it was a short talk, was just 10 megabases, base pairs, but we looked at lots of different ages, and it does change a little bit over time, the perspectives, but the pattern is the same.
Yeah, so the prediction was that with inbreeding, you will have a lot of variance in fitness, and so some population will be purged, and will have a higher fitness, but some will have a high fixation load and will probably go extinct. So do you think that the populations that we see now that are highly inbred and persist are a biased sample of just the one that persisted with inbreeding, and we lost all the others, which
Yeah.
doesn't mean that inbreeding is good for purging, but it means that that's the only one we can observe now.
Well, I think this is the problem with genetic rescue, is maybe we'll lose them all if we interfere with those populations that have survived. So, I mean, these are really extremely bottlenecked populations. So we wouldn't recommend this for populations where you might have multiple populations that have some chance of increasing variation, but for these really highly threatened populations, that's when I was suggesting they have survived, and they seem to show fitness that allows them to adapt. But no, it's not an easy question, obviously.
Unfortunately, we don't have time for any more questions, but definitely catch Barbara after the break, or during the break. So just point this at the computer when you want to change your slides. Okay? I'm going to introduce you. Okay, next up we have Ryan Chaffee, who is a PhD student at Cornell University. Ryan works on methods development of computational tools for wildlife management, and today we'll be discussing a model for conservation of animal populations experiencing habitat fragmentation.
All right. Okay. Thanks everyone so much for showing up today, and thanks to the organizers for including me in this symposium. As I was introduced, I'm Ryan. I'm a PhD student. I'm almost entering my third year. I'm in the lab of Jae-hee Kim at Cornell, and I'll be talking about one of the many, many projects that come out of the Kim lab. And the focus of my talk is modeling conservation of animal populations experiencing fragmentation. Okay, so to start off, it's important to note that in the Anthropocene, habitat loss is one of the leading causes of biodiversity loss across the globe, and landscape fragmentation can disrupt natural habitats and accelerate extinction for vulnerable organisms. And the image here shows an example of how landscape fragmentation can result in these isolated habitat patches that are completely disconnected from other habitats in the area. And modern genomics technology does offer precise genomic monitoring methods for conservationists, but the efficacy of this tech relies on the knowledge of when and how to implement it. And so therefore, it's vital that we develop a framework to understand how habitat fragmentation affects the genetic diversity and population dynamics of organisms in some landscape. And one such population subject to increasingly severe habitat fragmentation is the Maasai giraffe, which is pictured here. It is an endangered subspecies of giraffes in northeast Tanzania, and it's divided into a structured population in part by the natural barriers present in the landscape, like lakes and escarpments. And escarpments are just steep cliffs. And these escarpments separate the population into an eastern ecosystem, which is the Tarangire, and the western ecosystem, which is the Serengeti. And the western populations are in a protected national park, but the eastern populations are not, and they face encroachment from human settlements and inroads, as well as other anthropogenic pressures. And so therefore, we're interested in the Maasai giraffe subpopulations that are present in that eastern Tarangire ecosystem. And if we zoom in to the eastern region, we can see here that the ecosystem population is divided into 4 major subpopulations. And they're on this map in blue, green, orange, and pink. And while there may be significant geographic overlap in the extent of each subpopulation's range, it's important to note that the strong social structure of Maasai giraffe generally inhibits mixing between the subpopulations. So therefore, we can consider each subpopulation as a distinct deme. And the movement of individuals between demes, though relatively rare, is measured by our field ecologist collaborators, and they can track the movement with individual resolution. And now that we are able to classify this ecosystem as a subdivided population, we can introduce methods of studying its dynamics using population genetics principles. And the first step in our approach is modeling this system as a network. And to do this, we consider the definition of a network as a mathematical object. It's a directed graph, G, comprising a vertex set and an edge set. And the vertices, or nodes, represent the distinct populations. And the edges represent migration in the direction of the edge, and the edge weight defines the migration rate. And so here we see a simple network with 6 subpopulations in their respective edges, just as an example. And we can use this network to study how different processes affect population diversity. And the interesting ecological process we're going to explore today is habitat fragmentation. So a fragmentation process is represented as successive deletion of edges in the network and can occur with a variety of patterns. Intuitively, we can imagine a type of fragmentation like shown here, where we start with the initial connected network seen on the left, and that represents the natural state of some landscape. Then, when a road is built through that landscape, migration between some of these populations becomes either more difficult or impossible, and so those edges are removed from the network, resulting in a moderately fragmented stage like you see in the middle. And then if another road is built, like on the right, we remove even more edges and are left with a severely fragmented network that has many nodes that are completely isolated from other nodes in the network. So now that we know how to construct a migration network and model fragmentation on it, let's apply this principle to our giraffe population. And so to model the Tarangire ecosystem as a migration network, again, we first assign each of the subpopulations to a node in the network. And so what I'm showing here is a configuration that's approximately equal to the geographic orientation that you see on the map, but it's not spatially explicit. And each subpopulation has a relative node size that represents the census size of each population, the largest being that MRC population in green, which has a census population size of about 900. And then we can add the edge set, which is shown here. And that represents the migration rates between each pair of subpopulations in both directions. And you'll notice that there are no edges connecting to this population, gesture, LMNP, and that's just because Lake Manyara, which is shown on the map here, sequesters that population, so that there's been no significant migration measured between LMNP and other subpopulations in the Tarangire. And so for the purposes of this talk, we'll just focus on these three subpopulations that actually form a connected network. And so it's important to first consider that there are theoretical approaches to understanding how fragmentation affects the population diversity. And the general workflow is shown here, where we begin with habitat patches and extract the migration matrix, like I just showed in the previous slides. And we can apply this transformation using a system of equations that turns the migration matrix into the pairwise coalescent time matrix T. Once we extract the T matrix, we can then do some calculations to find values like the FST matrix or expected heterozygosity. And while this theoretical framework is very nice, it relies on some very stringent assumptions, such as neutral Wright-Fisher dynamics, equal population sizes, migration drift equilibrium, and symmetric migration. And all of those are very hard assumptions, and more importantly, they're not ecologically realistic at all. I already showed you that our subpopulations have different sizes, so even from that first glance, this theory doesn't apply very well. And so in order to circumvent the hard assumption by the theoretical model, we implement a simulation-based approach using SLIM. And using SLIM as a backend, we developed an eco-evolutionary simulator with the following characteristics. It's an agent-based simulation, so we can look at individual statistics across any point in time for every individual in the population. And we can apply overlapping generations, which is a much more complex population genetic scenario. We can then also model arbitrarily complex eco-evolutionary scenarios and impose that on our population of interest. And we can directly measure genetic diversity statistics at any point in time throughout the simulation. And using this simulator, we can also calculate the extinction risk of a given ecological scenario. If we run many, many, many simulations, we can count the number of replicates that go extinct within the demographics of the given scenario and estimate the probability that that scenario leads to extinction. And so using that simulation framework, here are some of the broader implications that can be applied to any organism, which is we allow for the effective monitoring of fragmentation, we can predict future genetic diversity declines, and we can also inform conservation and management regarding the remaining corridors that are present in a landscape that has been fragmented. So here's the protocol for our simulation, and this is general, even though you see some pictures of giraffes. And so firstly, a migration step occurs where individuals move between subpopulations in our landscape with some probability. And then we employ a reproduction step where we mutate and mix the genomes and produce offspring. And then we conduct fitness scaling based on the age of an individual as well as the aggregation of deleterious mutations within its genome. And then finally, each individual must pass a survival check. And the probability of survival is a function of both the individual fitness of the organism and for each individual, as well as density-dependent effects that come from the user-defined carrying capacity of the deems that you've put into the simulation. And so these steps repeat every single year, and they define the core process of the simulation. And so for as an example, we're using our giraffe to do a proof of concept. Here are some giraffe-specific ecological factors. Some have already been implemented, like sex-specific migratory behavior, where mature males continue to migrate through the landscape through their entire lifespan, but mature females only migrate once or 0 times in their life. We also have our deems defined by social structure rather than by geographic expanse. And some upcoming considerations are giraffe-specific mate choice behavior, seasonality and reproduction, proximity to human settlement, and also age and location-specific predation risk. And so the overall simulation demographic model is shown here, where we employ a 3-epoch approach. The first epoch is the ancestral population, which is a single large ancestral population, and then it splits into the eastern and western ecosystem. And so you see that the eastern ecosystem is then further separated into those 4 subpopulations I talked about. And just for the purposes of this, the western ecosystem is just a single population that's being modeled. And then from that time of divergence between the eastern and western ecosystem to the present day, we apply a given pattern of migration. And then once we reach the present day and move into the forward future predictions for our simulation, we can impose whatever demographic scenario we're interested in studying. So perhaps that might mean decreasing the migration rate or eliminating it altogether, which is represented by those question marks in between those populations that were previously connected. And so now that I've shown you what our model looks like, how do we know that it's a good model? We can validate the simulation model by comparing the output at the present day to the empirical data that's actually collected from this population. And so the data that we have access to are whole genome and mitochondrial DNA sequences from 100 giraffes in northeast Tanzania. And those first 3 numbers for the 4, 29, and 36 are from the eastern ecosystem, and the remaining 31 are from the west. And then using these sequences, we can then extract some genetic measures that are of interest. Specifically, today I'll talk about FST and FROH, where FST is the fixation index that measures genetic dissimilarity. FROH is the inbreeding coefficient that measures inbreeding as a function of runs of homozygosity present in the genome. And so these are the actual present-day values that have been extracted from the empirical data. And then when we look at a comparison to what the simulation outputs, here in these charts, you're seeing 100 replicate runs, and then the black line through them is the median value. And that red dashed line is the present day, where the values, we can compare the value from the simulation output to the actual data collected. And you can see that for both FST and FROH, the predicted value from the simulation is very, very close to that to the value that is shown in the empirical data. And so this sort of validates our simulation as a good approach to modeling this population. And so if we want to fragment this population and ask what happens when we do so, here's an example of what I'll be talking about, where if you look at this map on the left, I've highlighted now a gray line, which represents a tarmac road that was recently built through this landscape in the past 10 years. And what this has done is it's isolated that MRC population, the one in green, from the other 2 populations in the Tarangire. And so what we have now are 2 different cases, where initially these were connected and now they're fragmented, and the 2 corresponding networks are shown here. And so what I'll do is compare some of these genetic diversity measures for specifically the MRC subpopulation. When we look at FST for the MRT subpopulation, in the connected case, we expect FST to remain relatively low from the present day onward. But when we fragment, like we did with that tarmac road, we see that FST gradually increases to much higher levels. When we look at FROH, we see a similar pattern where it was expected to remain the same through the connected case, but when we fragment, the FROH value begins to increase, showing that there's an increase in the level of inbreeding in this population. And lastly, I want to show the population size trajectory. So there's many, many lines here, but what the takeaway is that in the connected case, that MRC population only went extinct in 5% of replicates. But once we fragment it, it now goes extinct in 22% of replicates. And if we look at the overall extinction risk for this entire network, the overall extinction risk, again, is 5%, which is, in this case, it's all 3 subpopulations go extinct in 5% of replicates. But when we fragment MRC, the entire ecosystem goes extinct in 8% of replicates. And then lastly, I'll ask, what is, if we fragmented the ecosystem to a severe degree, where we completely removed all of these edges, what is the outcome? And we see that the extinction risk increases from 5% in the connected case to 20% for the entire ecosystem in the fragmented case. And so in conclusion, I've shown you that we predict a 4-fold increase in the extinction risk for Maasai giraffe subpopulation due to recent habitat fragmentation. And our simulator predicts the impact of fragmentation on survivability, offering insight into informed ecosystem management. And with that, I'd like to acknowledge my lab and our collaborators. Thank you so much for listening.
Just point at the computer when you go to change slides. Just introduce you. That was a great talk. For our last talk of the methods section of our symposium, we have Troy LaPolice, who is a PhD student at Pennsylvania State University. Troy's research uses interdisciplinary methods in modeling and genomics of past and future populations. And today, Troy will be telling us about the digital reef.
Hi, everybody. My name is Troy LaPolice. I'm at the Pennsylvania State University in Dr. Christian Huber's lab. And today I'm going to be talking about how we can take insights from computational agent-based modeling to inform coral restoration efforts. And I'm going to be talking today about a Caribbean coral species complex, which unfortunately is critically endangered. And I'm going to be talking about how we can bring this into the digital space with the idea of bringing it back in real life. And first, to provide some background as to why we care about corals and coral restoration, it's because coral reefs are an extremely important system. Reefs provide coastline protection by dissipating potentially damaging wave energy before it can make landfall and erode the coastline. And from a biological perspective, reefs support biodiversity. It's thought that reefs support 25% of all ocean species. And if we start to lose these corals, it's estimated that we could lose 50% of all tropical fish biodiversity. And unfortunately, that's what's happening. It doesn't take too long to do a quick Google search and find many popular news articles talking about mass extinction and mass die-offs of coral reefs throughout the globe as a result of various anthropogenic factors. And so what humans are trying to do to restore these decimated populations and try to bring them back are put together a series of restoration methods. And one of these restoration methods is outplanting. And so in outplanting, what restoration practitioners will do is they'll take advantage of coral's natural ability to asexually reproduce via fragmentation by taking healthy pieces of corals from nursery-grown populations or other wild populations and planting them on vacant reef substrate with the idea that it can grow and mature alongside other outplanted corals, with the ideal outcome being a healthy and independently reproducing reef. But you can imagine that to get this ideal outcome, it takes a lot of time, takes a lot of money, and it takes a lot of effort. And so with the populations massively dying out, there's a clear need for optimization of our restoration efforts, both from a biological perspective and maximizing the amount of bang we can get for our buck, if you will, in terms of the economic optimization. And so what our study system, how it fits in here is we look at a Caribbean species complex in the acroporid, the Acropora palmata, Acropora cervicornis, and Acropora prolifera. And palmata and cervicornis are two species that are commonly outplanted in the Caribbean, and these are parental species that can hybridize to form their F1 hybrid Acropora prolifera. And some evidence has shown that prolifera can backcross with its parental species and potentially has limited success in creating F2s, but it can backcross with its parental species. So this is what these corals look like in the wild. These photos were taken by my collaborator Colin Howe on a dive in the Caribbean. These are branching corals that live in the shallow reefs, and you can tell palmata has this more flat structure, cervicornis is more tubular, and prolifera has this intermediate morphology. And for millions of years, these were dominating the shallow reefs of the Caribbean alongside one another, but unfortunately they've experienced a really serious population decline with a near complete loss in many areas of the Caribbean. And so where our project fits in is we want to understand how different outplanting regimes affect the integration rates and the species dynamics in this population. And so what you can imagine is if restoration practitioners are individually planting fragments along a reef landscape, you can imagine they have a lot of influence over the architecture of the reef. And so one of the ways they have influence is in the species that they choose to outplant. So say they plant a more balanced regime where they're planting half and half, or if they're favoring one species over the other, under the idea of, let's say here, Cervicornis is the fastest growing, so it can cover more reef space quicker. What is the impact of this on the population dynamics and the integration? And also we can think in three-dimensional space. So while these corals live in symmetry, it's known that Palmata prefers the more shallow end of their shared range, with Cervicornis preferring more deep. So in how we use that in our restoration, are we planting them mixed together where they're all competing with one another for the reef landscape and resources, or are they independent where they're only competing within species? What is the impact of this? And chiefly, we're interested in understanding how the F1 hybrid fits into this. Because it can backcross with the parental species, there is some concern that the increased gene flow between the parentals via integration in backcrossing could lead to a breakdown in genetic diversity and ultimately lead to genetic swamping within the population and reduction of local species diversity. And so what we do to try to understand the impacts of these things are we impose an agent-based model. And some of the other presenters have talked about using SLIM in this way, but just to give a little introduction on what an agent-based model is, rather than modeling a system as a single large entity, it models individuals and their interactions. So let's say we take traffic, for example. We can model traffic as a blob of cars moving down the freeway, but we know it's a little bit more complicated than that, with individual drivers making decisions. One driver changes lanes, everybody hits the brakes, and it leads to this complicated system. And each individual driver makes decisions that lead to traffic as a whole. And so we take this into the coral sphere and we look at individual behaviors of each coral, so their spawning events, their mortality, and their competition with one another. And at the simulation of the micro level, we get a better picture of the macro level. And so this approach has been applied in other fields, in population genetics and biology, but rarely in coral biology. And the models that have done this in corals, they rarely provide a genetic component, as our model will show in a little bit here. And so in our model, we feature the 3 Caribbean species that I spoke about, and we impose literature-based parameters which govern various aspects such as the sexual reproduction, asexual fragmentation, their larval mortality, their adult mortality, the reefscape that they live on, etc., as well as many other parameters that work together to make this an accurate representation of a really complex system. And most importantly, each coral in our model has an independent genome. Its individual genome has the ability to, they're generated via sexual recombination of the parentals and clonally from fragmental reproduction. They have realistic recombination rates and chromosome lengths based on sequencing of these species. And what this importantly allows us to do is answer genetic questions about this system. And so we can look at both the species dynamics and these questions about population genetics. So what we've found from these simulations is starting with overlapping niches, so we're planting them within competition with one another. The idea they live in symmetry in the wild, so we're going to plant them that way and see how that happens under various outplanting regimes. So if we're focusing on more balanced or favoring one species or another, we get these results. And so what these plots are, they're a little complicated, but I'll walk you through them here. These are faceted by the outplanting regime. I don't know if you can see my cursor there. But there we go. So they're faceted by the outplanting regime, so the number of species that we're favoring in the outplanting plan or a more balanced, and what it represents at time 0, an outplanting event, and tracking this throughout time. So projecting into the future how these outplanting events, the trajectory of them. And what each line represents is the species that we've talked about throughout the presentation by their color scheme that you've seen throughout. And this is the total number of individuals in each species over time. The secondary y-axis on the left, it represents the introgression. So what this is the parental ancestry that's introgressed from one parental species into another via backcrossing with the hybrid. So Cervicornis ancestry that has introgressed into Palmata is in the pink color, and the cyan is Palmata ancestry that has introgressed into cervicornis. And so what we see here is that it becomes readily apparent that when we have this outplanting regime, the species ratio that we plant makes a big difference here. So the species that's presented or planted in a higher number, it quickly becomes dominant in the population, and it does well and generally outcompetes the other species. They don't go extinct, but they struggle to gain foothold on the reef as a result of the primary species taking up all of the space. We also see here that the hybrids are able to outcompete the minority taxa in a lot of these circumstances. And we can see when we have 50-50 outplanting that all the species end up doing about the same over time. Initially, the parentals do better, and then the hybrid can gain some ground here. From an introgression perspective, what's interesting is that it's not particularly high across the genome in terms of the percentage that we're seeing of the parental genomes that have introgressed between species. And interestingly, when we favor one species over another, we see a really stark contrast in that the introgression is unidirectional. So it's from one species into another, but not the other way around. And we see that this doesn't happen when we favor that, or when we don't favor anything in a more balanced regime. But we can see that when we do, it's typically from the minority taxa into the majority taxa. But this changes when we look at partially overlapping niches. So in this, we plant a little bit across the depth gradient with 2 preferred niches and an intermediate habitat where they're competing with one another for space. And we do the same with different species ratios. And these are those same plots. And we can see that the story becomes a lot different. The parental, if you look in the first few years, it seems as though they're going to end up being the same. We see the dominant taxa becoming dominant, the parental species staying low. But over time, we see a very different dynamic where as the minority taxa starts to grow its population size within its preferred habitat, it's then able to get to a population size where it can start competing for the intermediate habitat. So that dominant species, it takes over its niche and the intermediate, but eventually it becomes in a position where it's competing with the other species, and it leads to this equilibrium over time. And we can see that all of the species do well here, with the hybrid doing slightly better, but in general they all reach an equilibrium as a result of this, which is very different from the previous slide. From an introgression perspective, we see that the unidirectional aspect is gone here. We can see it's a little bit biased in one direction or the other on the extreme ends, but we see slightly higher levels of introgression. Now, on the whole, it's still not a super large amount, and so because we don't see a ton of introgression, it might indicate that the ability for genetic swamping might actually not be quite as high as we expected. But what this should all go to show is that it's really important to consider the desired outcome when selecting an outplanting regime, because outplanting regimes affect the species dynamics. And so what we've seen so far is that the most important parameters in determining both the species and success and the directionality of the gene flow are the initial outplanting ratios and the degree to which the coral niches overlap. And this is when all things are even. So one of the things that we're going to consider going forward is beneficial alleles and how they affect the system, the potential for evolutionary trade-off, as well as the potential for adaptive integration. So the beneficial allele being in one parental species and being able to introgress into the other via the hybrid, as well as dynamic bleaching, climate projections, and variable reproductive success within the hybrid gametes. So with that, I would like to thank my lab, my collaborators on this project, Christian Huber, Colin Howe, and Nick Locatelli, as well as my funding, Penn State, and the organizers of this symposium and this event. So with that, does anybody have any questions? Thank you.
So I was wondering, how do those tiny pieces of coral or maybe juveniles get to grow in an environment where the water is warming and the already grown knobs are dying?
That's a great question that restoration practitioners are constantly wrestling with. It's hard. The corals, they really struggle, and a lot of these restoration plans, it's a constant battle. And some of the ways that people are working are to in nursery stock, increase resilience through genetic breeding and artificial selection for warming-resistant alleles. And really, lots of people are attacking this really complex problem from a lot of different directions. It's going to take a really collaborative and interdisciplinary effort that involves the restoration practitioners, geneticists, and everybody involved to come to a solution, because it's really fighting an uphill battle, like you mentioned. The oceans are continuing to warm, and we're doing damage control as best we can, but it's going to take a really big effort to get this to happen. I think I'm maybe out of time here, but you can find me afterwards. I'm happy to keep talking. Thank you.
All right, everybody's starting to trickle in for the next installment of our session of conservation genetics across multiple species. We had a really nice group of talks this morning that were more focused on methodological aspects and methods development for conservation genetics. And now with these next two sessions, we're going to move into work that deals with more empirical evidence. And so our first speaker is Jarrod Sauve, who's currently a postdoc at the University de Quebec in Montreal. And he's interested in human-affected wildlife populations and is currently working on studying evolution and phenotypic change in conservation breeding programs. So happy to turn the floor over to you, Jarrod.
Thank you.
Awesome. Thank you, Chloe, for that nice introduction, and thank you both, Chloe and Ruth, for organizing this symposium. I'm excited to see lots of the talks. So today I'm going to talk a little bit about genetic adaptation in zoos, and kind of where there we go, got it. Kind of where I come from on this angle is I started a lot studying wild populations where we had these big questions about how fast adaptive genetic change was, particularly trying to answer questions like, can we predict population persistence in face of things like climate change, habitat change, different changes in the environment in general. And for today, I'm going to focus a little bit more on this kind of management side. So asking questions about if we're trying to do ex situ breeding and breeding animals in captivity and releasing them into a wild setting, how might evolution in this setting affect this aspect of conservation management? And so I'm coming at this from a quantitative genetics angle, where I'm interested in this particular parameter, the additive genetic variance in relative fitness, or Fisher's fundamental theorem of natural selection. This states that the per generation proportional change in mean absolute fitness caused by natural selection is given by the additive genetic variance in relative fitness. And kind of my way that I like to try and explain some of this is just using this simple diagram with acorns. So on your left there, we could imagine that all these black acorns are potentially parent acorns. On the left, we have each one producing one offspring over the entire life. So we have no variation in fitness on the left here. On the right, we have variation amongst the different parent acorns. So it's in this situation that we might expect some evolution by natural selection. So there's some important things about measuring the additive genetic variance and relative fitness. We need quite a lot of data. We're going to need relatedness among individuals in a population, accurate measures of fitness over their lifespans, and importantly too, I think we need to try and capture some of these confounding factors like common environments, gene flows, maternal environments, to try and get a better measure on this. And really the inspiration for the work I'm presenting today comes from recent results in the wild in 2022, where they went out and looked at 19 of these long-term studied populations and measured the additive genetic variance in relative fitness. Just the graph on the right there is showing the posterior estimate for each of these species. And kind of one of the main conclusions they got from this is that it seemed that adaptive genetic change was occurring 3 to 4 times faster than had kind of previously been thought. What I'm hoping to do is shift this, and what I'm going to present today is a little bit at looking at zoos. There's a lot of zoos that are really interested in contributing to conservation management now. There's kind of been this shift in focus towards increasing conservation activity. We also have mandates now in the biodiversity plan. Target 4 specifically mentions the use of ex situ conservation breeding to try and supplement some of these populations. But in order to do this, we probably want to get a handle on how much evolution might be going on in some of these breeding programs. We know that from different studies in different species, these are just a few examples here I've pulled, we have evidence of trait change. So in Hubera bustards, we see over generations in captivity a change in the mean number of eggs laid. Something recent work that I've been working on in eastern loggerhead shrike, we see some morphological changes over time in these captive breeding programs. We also know that when we release individuals or translocate individuals, they tend to perform poorly compared to their wild counterparts. So just different taxonomic groups on the y-axis here. And if you can see kind of the dashed black line, everything to the left of that suggests that in an introduced case, the species are performing more poorly than their wild counterparts when they're released. So it kind of leads to the potential question, could adaptive genetic change in captivity be affecting reintroduction success? And what I'm trying to tackle today is just whether or not we see evidence of adaptive genetic change in these captive breeding programs is kind of the first part of this question. So again, we're going to need lots of data to get a measure of this. We're going to need relatedness among individuals and accurate measures of fitness. Thankfully, there's really well-suited data sets in the zoo world that can kind of handle some of this. So all of this project, I've pulled data from Species 360, which is this very large database of 10 million animals that they have records on things like pedigrees and stud books. And it's been pointed out in the past that these are quite well suited for these quantitative genetic studies. To give you a sense of the data sets that we actually worked with, we ended up going with 31 different species where we were able to get these big pedigrees. It's a total of 2,500 years of data. The longest maintained records in some cases go all the way back to 1877. Some of the shortest maintained records are as early as 1994. And in total, the data set that we analyzed has 260,000 individual animals in it. These are just a few examples with the pedigrees. All these kind of gray lines are connecting parents to offspring for particular species. But we had a lot of data, and it was, I guess, a better situation than we thought when we first went into this project. For the methods, we modeled absolute fitness using generalized linear mixed effect animal models with a zero inflated Poisson distribution. There was a lot of work done by Bonnet et al. And Morrissey and Bonnet to kind of get a handle on how to model fitness appropriately in these models. So we used what they did in the wild populations. We have some fixed effects that we're adding into these models and random effects. The kind of major difference that we have is that we have from the wild populations is that we also have birth facilities. So these animals are often housed in multiple different institutions. So that's a slight difference from the wild where they're often monitoring kind of one population. So we have that in there as a random effect. And what we found when we got this estimate from all these different populations, which to me was quite surprising at first, is that rates of adaptive genetic change were remarkably similar to this recent estimate in the 2022 study. So we have our estimates of the additive genetic variance of fitness on the left here, and Bonnet et al.'s estimates from 2022 on the right. If you look at a lot of these numbers, they're quite similar across the board. So we have kind of similar rates of adaptive genetic change occurring in these conservation and zoo settings. Just to show you what that looks on a graphic here, we have these ordered from kind of our lowest average variance in additive genetic variance, the top left going back down to some of our larger estimates. These ones are colored based on the lower credible interval being greater than 0.001. It's roughly the mode of the prior that we used for these models. And then we also have the darker purple for when it was greater than 0.01, which indicates, according to some of the previous studies, quite rapid adaptive genetic change occurring in these populations. So it kind of brings up lots of questions, like why do we see additive genetic variance in fitness in some of these conservation breeding programs? A lot of them are trying to follow these minimizing mean kinship methods that should reduce variation in fitness. I think you often have variation in the logistics of actually implementing these kind of programs, you can imagine that some species it's going to be easier to move an individual to reduce mean kinship versus others, like for example a rhino or something is a lot harder to transport. I do think it's potentially not surprising given that the zoo environment is still quite different from many of the wild environments these animals are experiencing, so it's perhaps not surprising. I think an additional factor is that many of these populations have quite complex management histories. Some have been started and managed solely with a purpose of conservation breeding, while others have, if you think back to that 1877, they probably weren't thinking about managing these species based on evolution back at that time. So we have this variation in terms of the management and the history of the populations that likely could contribute to the variation we see among these different populations. So what does this kind of all mean? We have potentially evidence that we have adaptation to captivity or the captive environment occurring. We have evidence of evolution by natural selection, which could suggest that the average fitness is increasing in captivity, which maybe is a good thing if we want to be producing in animals in this kind of ex situ environment, but it could come at a cost if we're thinking about releasing them, right? If they're adapted to the captive environment and not the wild environment in which we're trying to release them into. We don't want to be accidentally kind of domesticating some of these species. And I just have this graphic on the right here. You can see the cursor here. So this is just to kind of put in context kind of a back of the envelope calculation that Bonet et al. Did in 2022, where on the y-axis we have our population size relative to generation 0. We have on the x-axis the number of generations in the population, and for each of these lines we have a different additive genetic variance of fitness for that particular population. And what they imagined is if you have some sort of environment that reduces fitness in the first generation, for these different additive genetic variances in fitness, how quickly does your population kind of rebound for this particular value? And for the estimate we got in our study, we have something in between these two blue lines here. So within 10 generations, kind of potentially impacting the demography of these species here. I think it's really cool in the sense that there's lots to dig into with these data sets. We have fitness variation among mothers, facilities, and cohorts that I didn't talk a whole lot about today, but again, I have the same graphic with the different species and these different variance components, potentially very interesting to dig into to figure out, why do we see variation among facilities or mothers in these fitness estimates. And for my main conclusions, I think this points towards genetic adaptation likely occurring in zoos and conservation breeding programs. We probably need to determine exactly why we see this signal in some populations and not others. Is it a management thing, an environment thing? I think our results potentially provide a starting point where we can pinpoint species that, where we see this high measure of additive genetic variance and the ones that we don't, trying to dig into why exactly that is. Potentially, in the future, a tool for managers, does their particular population show signals of adaptive genetic change that they're working on? And I think there's lots of open questions, so my results don't tell us what selection might be acting on, right? Just tells us that, it could be one trait that's heavily selected for, or many, many traits that are all changing a very small amount. And then trying to figure out what the consequences are for reintroduction. Is this having a big impact on our ability to reintroduce species? With that, I have lots of different people to thank and funding sources. And just while I take questions, I wanted to highlight some relevant talks and posters. So my colleague, Hannah Thompson, is giving a talk on variation in fecundity in the zoo setting at poster E12. And on the last day of the conference, I think many of you will also be interested in this Evolution in Zoos and Conservation Breeding Programs symposium that we have. So please attend if you can. And thank you. Yeah.
Species that do show a rapid response in captivity, do we see that the longer they stay in captivity, the worse they do when they're reintroduced?
So I haven't looked at that yet, but it would be an interesting thing, to check out if it's been a longer time. I would imagine, it seems to make sense, but yeah.
Do you know, just the breeding value?
it's the next thing that I'm just about to do, is to see if it actually does increase, if we see that change over time. Yeah.
You alluded to the mating system design for maximizing, regaining variance.
Yeah.
Maybe you're just
Increasing the variance. Yeah. Good question. Yeah.
So is it necessarily an adaptive response in captivity, or could it be a relaxation of the
Yeah, I mean, I have to think about that for sure, but maybe we can talk. Yeah. One versus the other. Thank you.
Thank you so much, Drew. Next up, we have Dr. Ellika Faust, who's currently a postdoc at EAVAG, the Swiss Federal Institute of Aquatic Science and Technology. Ellika is interested in studying evolutionary and ecological processes in aquatic systems and their conservation systems. So happy to pass it over to you. This is a slide changer.
Okay.
If you pointed at the left.
Perfect. Thank you so much. So thank you, first of all, for letting me come and present here. So I'll be talking about freshwater systems today. And during my postdoc, we've had this idea of trying to establish this kind of framework for working in these systems. And first, I thought I'd just highlight some of the things that makes them a little bit different. So first of all, freshwater habitats are really, really fragmented. So we heard earlier about fragmentations when you have a road. Here, you literally cannot cross land. So you really have to just stick to these small little pathways to be able to connect. So you have really limited connectivity as well as dispersal, and you end up with these really small and localized populations. And this is kind of then exacerbated now in the Anthropocene. So you have dams and hydropowers who kind of limit or makes more fragmented habitats and limit this connectivity that we have. But we also have humans who move, for example, boats between different lakes. So we introduce new invasive species to a higher rate. And this, of course, also affects the localized and small populations, but we also have, for example, direct exploitation in form of, for example, fishing pressure. And in addition to that, we have pollution, such as chemicals and nutrients, hydrological alterations, such as channelization or flow regulations, and of course, climate change. So increased temperature, but also increased precipitation has major effect of where these organisms can live at all in these different systems. So they are really kind of in danger in this way, and we also know this because if we look at global biodiversity loss, it really is absolutely major in the freshwater systems. So since the 70s, it's been expected to have lost around 81% of freshwater diversity. So it's really, really happening at a very rapid speed. So this is why we want to work on freshwater biodiversity. I'll move that there. And what we really wanted to do was trying to use whole genome sequencing to assess some basic parameters, so population structure, population diversity, and population vulnerability. And as we were going, we kind of realized, okay, this aligns actually really nicely with the GeoBond's genetic EBVs, which I would assume most of you are familiarized with, by Hoban et al. In 2022. So genetic divergence, diversity, effective population sizes, and breeding. So we wanted to try and focus on these different aspects and see how would we assess this with a similar pipeline within freshwater systems for very different taxa. So we use these two very different study systems. So we have the European grayling, and you have the duck mussel. So European grayling lives in rivers and requires different types of flow regimes for different types of its life stage. The duck mussel lives in small ponds, lakes, or maybe slow-flowing waters. Of course, you can imagine they also have very different ways of how they even disperse in the system. So they're really quite different. They also have some similarities, and that is that they're very widespread across Europe, but they're also very much declining in a lot of different areas. And that's often due to a lot of these major biodiversity loss drivers as we know them. And one of the places where they're also really declining at a quite rapid speed, and some of them have even been lost, some of the populations, is in Switzerland, which is where I'm working. And just for those of you who might not be familiar exactly where Switzerland is, it's a tiny little country that's landlocked in Europe. It's about a quarter of the size of Quebec, so it's not very big. But it also has this really large alpine range of mountains which separates these water systems. So the waters actually flow into 4 different seas, so the North Sea, the Mediterranean Sea, the Adriatic Sea, as well as the Black Sea. So even though they geographically might not be that far away as a bird flight, they actually might have been separated for a really, really long time. So I'll kick off with the European grayling. So here we started off actually working on existing samples. So we sequenced around 200 different individuals from across the different catchment areas, and I will kind of go straight on into the results that we found. So here you can first see just a simple PCA, and you can show that we have three very, very distinct populations. So first of all, you can see in the south, so in the end, let's see if I can, down here, so this is then south of the Alps, so they're really highly divergent from the other ones, as well as just two individuals, which was unfortunately the sample size that we had up in the northwest. Whilst all the other samples, even though they're from different catchment areas, all cluster together. And this is also more than 50% of the total variation that we have in our data set, so it's really, really major divergence. We do see some more differences within this little cluster down here when we look at the next PC, so there we can actually start seeing how the different catchment areas start to differentiate, and we can see how the different populations actually do also have some differences there, but it's much, much smaller compared to these large differences we've seen between the north and the south of the Alps, for example. And what's more is that we didn't just look at these wild populations, we also looked at hatchery populations. And this is because this species has been artificially stocked for about 50 to 70 years. And we also know from previously that some of these sources are not always in Switzerland. And this is also something we can very clearly see here, as we have these three different hatcheries down here who were really highly divergent from any of our other populations that we can find wild in Switzerland. And the divergence is also really high. So the divergence between these three different clusters, as well as these three different hatcheries, is between 0.3 and 0.6 FST. So they really are very, very different, whilst the rest of Switzerland looks basically homogeneous in comparison. But if we do dig a little bit deeper, we do actually see that we have the same kind of pattern as we saw also on the PCA, that we do have roughly 7 wild clusters that do correspond pretty well to the different catchment areas that we also do have in Switzerland, as well as these 3 different hatchery clusters. And in terms of diversity, we can also see that these 2 very divergent populations, so again, the one in the south and the one in the northwest, have much higher heterozygosity and also nucleotide diversity in comparison to our other populations. And not so surprisingly, they also have much less inbreeding. And we do have some hypotheses about these, and that's because we do actually know that there are sister species that have previously existed or may still exist to some extent south of Switzerland as well to west of Switzerland. So this could actually be an explanation for why they have such high diversity in comparison to what we see elsewhere. And if we look at the effective population size, this is also kind of what we were expecting. So we have really seen a major drop in the last 25 generations for all the different clusters, most of them even within the last 5 generations. And only 1 out of 6 clusters that we could test for have any above 500, which is, as you know, the new indicator that we're reporting to the CBD. So in conclusion, we have 7 very divergent populations. There's a high diversity in the south as well in the northwest, and all of the populations are really decreased in their effective population size. And 3 out of our 6 hatcheries that we tested do not have grayling from the country, or they're local to the area. Now for the duck mussel, I first want to just start by highlighting that a lot of this work done by my collaborators, first Sauve Gwyneth, who assembled the genome, and then Julie Conrads, who was a master's student, who's now starting her PhD, did a lot of the population genomics work. So for the duck mussel, we actually went out in the field and sampled, and it turned out that we did not just get the Anodonta natina, the duck mussel, but also the 2 sister species, because they're not super easy to kind of disentangle. But we still managed to get most of the different catchment areas, also for the Anodonta natina, as you can see the different red areas here. But it turned out that this one down here, even though it has the same CO1, is actually highly divergent from the other ones. And if you look at this tree, you can see that where this arrow is here, that the divergence between this population and the other Anodonta natina north of the Alps is almost the same as what we have for the 2 different sister species. So similar to the grayling, there is massive divergence between the north and the south of the Alps. And the same thing we also see for the population structure, so it does seem to follow the catchment areas. I can point out, so the red, orange, and yellow areas, they do actually connect in some places, so they're not completely isolated, but they're still part of different catchments. But in addition to that, you can also see that a lot of them do seem to cluster very closely together here, And that is because they are from often very small little ponds or lakes that might have really, really poor connectivity. And if we just look within one of the different catchment areas, so the Are, you can see that all these different ponds are really quite different. So it's not just that they're different catchment areas, but they're also different within the different catchment areas. But even though there are in these little ponds, there is still connectivity, and we can actually see that there is also isolation by distance. So somehow they're still able to actually migrate out of these ponds and do share some gene flow as well. And when it comes to genetic diversity, we realize that size really matters. But was that me? But the size matters not in the terms of how big the mussel is, so they can get quite large, but that's not what I'm talking about, but actually the size of the lake or the pond that they live in. And in relation to the talk that was earlier this morning, it talked about the area size that you sample and genetic diversity and the correlation with that. This is not because we sampled more of the lake. We still sampled roughly the same area, it's just the lake itself is larger. So the population simply can be larger and have more diversity and more exchange. And not so surprisingly, again, the inbreeding had the opposite effect, so then smaller ponds and lakes have a higher inbreeding. So in summary, we can say that there seems to be an existence of maybe a cryptic species in the south. There's very clear isolation by distance, but almost every pond is still different. Genetic diversity is positively correlated with size and the pond and the lake, or inbreeding is also negatively correlated with the size of the pond and the lake. So kind of to take home from this is that in general, the population south of the Alps may be more accurately defined as species as subspecies, although maybe we don't want to talk about species at all anymore. I would love if we just talk about population diversity, that would be great. And water body size is highly correlated with diversity. And I would love to look for this also for the grayling, but it's much harder to assess water body size for river systems and ponds and lakes. Catchment areas are really good proxies for metapopulations, but I would say for both the grayling as well as the duck mussel, we do say that almost every site that we measure is genetically divergent from the other. So really conservation efforts really need to be targeted on habitat preservation, but more than anything, maintaining connectivity. Because all these dams and hydropower plants really limit this and completely isolate these populations from each other, because they cannot cross land, how much they ever try. And with that, I would like to acknowledge a lot of different people who have helped me both get permits to sample all of these different things, as well as working on the data. And also, thank you so much for listening. Potentially, yes. So, I mean, you do also have some lakes are really deep, so some of them can live really far down. So that could be a possibility, absolutely, that they can also be locally adapted to a different environment. So we have seen that we looked a little bit at some correlations, and we do see that they do correlate a little bit with, for example, temperature of the lakes. For example, the smaller ponds will heat up significantly more than any larger lake would, for example. Okay. Then thank you very much.
All right, so we still have a minute or two here before we started, so I'll introduce you very slowly. This is Guillermo Friis, who is currently a postdoc at the Royal Botanic Gardens in Kew in the UK. He is interested in the origins and maintenance of biodiversity, the genomic basis of phenotypic variation, and in general, adaptive processes across anthropogenic and restored habitats. And today he's going to speak to us about the genetic consequences of tree planting. So,
sort of wait.
2 minutes, I feel like, yes, I should give people a chance.
Yeah. Okay, thank you, Chloe, for the introduction, and thanks you all for attending this presentation, which we title Genetic Consequences of Tree Planting versus Natural Colonization, Implications for Restoration Programs. The government of the United Kingdom has committed to establishing 30,000 hectares of new woodlands annually from now until 2050, aiming for net zero emissions of greenhouse gases by that time. However, there is an important gap of knowledge about the genetic diversity of the planted forests when compared to naturally occurring ones, and about the impact that planting may have on resilience to pests and diseases and to climatic change. Afforestation strategies in the UK to meet conservation goals can be broadly classified into two main types. Afforestation pipelines based on natural colonization, in which tree communities establish through natural dispersal into former arable and farming lands, and afforestation pipelines based on active planting using commercially produced trees. Now, both of these strategies involve different stages and processes that can affect the levels of genetic diversity of the resulting populations. Parental trees in wild populations may arguably represent wider genetic pools and be better adapted to target sites than those produced in sea orchards. In turn, factors such as climate, predation, incidence of pests and diseases can reduce the levels of genetic diversity through differential mortality across age cohorts. Conversely, planting protocols that integrate multiple seed provenances and aim to maximize germination and survival through highly favorable growing conditions can actually result in more diverse populations than natural ones. And it is possible that these populations, through natural selection, are better able to keep pace with emerging stressors. However, seed collection, germination, nursery cultivation, and planting itself can introduce genetic bottlenecks and selective pressures we are unaware of. In this study, we test these competing hypotheses focusing on three specific age cohorts in natural colonization pipelines, parental trees and seedling and sapling colonizers, and three corresponding life stages in planting pipelines, parental trees in sea orchards, seedlings in nurseries, and recently outplanted saplings. More specifically, we focused on pedunculate oak and silver birch to implement a multidisciplinary approach aiming to, first of all, test for differences in between wild and commercial bioclimatic niches. We aim to test or to assess to what extent seed sources forests represent the climatic space that they occupy, represent the wild distribution of the species for a set of UK native species. We also compare levels of genetic diversity across age cohorts and life stages in natural colonization and planting pipelines to evaluate the impact that they may have in the resulting populations, and thereby identify potential bottlenecks and selective pressures using whole genome sequencing data. So moving on to the results, to test for differences between wild and seed sources bioclimatic niches, we combined remote sensing climatic data from commercial growth sites for 39 UK native species as registered by the Department of Environment in the UK. Here I'm showing you 4 as an example, and corresponding wild distributions as available on the Atlas 2020. So what we did was to map the available location of seed source forests as shown in the left panel, and we compared them to the wild distributions using principal components analysis and permanovas. So you can see in the right panel that the climatic space occupied by the seed source forests was consistently smaller than the climatic space occupied by the wild distributions. These differences were conspicuous and significant for the great majority of the species that we tested, and this result reveals that there may be substantial loss of adaptive variability when seed collection is performing for planting in the UK. Now, for exploring patterns of genetic diversity, full genome sequencing was conducted for around 25 individuals for each one of the mentioned cohorts and life stages. We sequenced, or we sampled 4 wild populations in the case of natural colonization pipelines and 3 planted populations per species, that is, for planting afforestation pipelines. We also sequenced, well, in addition to 2 trees from 2 nurseries and 1 orchard. We also sequenced representative populations of ancestral UK forests, matching the seed source areas to enable comparative studies. In total, nearly 600 genomes per species were sequenced, and nearly 30 populations were included in the study. So we tested for differences in genetic diversity, the genetic diversity levels potentially arising from genetic bottlenecks using several genetic indices, and based on hundreds of thousands of high-quality independent SNPs. As you can see in the figure, we computed expected and observed heterozygosity, nucleotide diversity, coefficient of inbreeding, allelic richness, and private allelic richness. And truth is that low disparity was found in most of the cases in these indices, even when comparing groups and populations belonging either to natural colonization sites versus planting afforestation pipelines. This seems to indicate the absence of strong genetic bottlenecks in planting populations, and also rules out the hypothetical higher levels of diversity in nursery and planted populations, potentially deriving from multi-origin seed batches and better growing conditions. Relatively divergent values were found only in private allelic richness. You can see in the figure there that colonizers for both species, oak and birch, showed higher values than the rest of the remaining groups. And this may indicate that incoming gene flow from distant populations may have an important role in shaping the levels of genetic diversity in the young colonizers' populations, which is important because this may indicate that more local standard genetic variation upon which selection may act happens in natural young colonizer populations. Well, maybe you're wondering about that big value there for a parental tree. That's a parental population that we learned too late that had been managed decades ago and some planting with foreign trees. It may have happened, so it's not at the point that we're going to use anymore. Anyways, we further examined patterns of genetic diversity by computing co-ancestry scores using the program ChromoPainter. Results were roughly similar for oak and birch, so I'm only presenting here those for oak. This is a heat map showing pairwise and within group average levels of co-ancestry, and there is a number of patterns that are of interest. First of all, we have, you can see that, right? Yes. First of all, we have these 3x3 squares along the diagonal in the upper part of the heat map, which basically represent higher co-ancestry values in between parental trees and colonizers in natural populations, which we expected. We also found that some of the highest values of co-ancestry happen in planted and nursery populations in within group comparisons, which reveals that, at least from some cases, these trees have a common provenance. And third, we realized that between group comparisons involving natural colonization sites generally had lower values of co-ancestry compared to those involving planted or nurseries. And this suggests that some degree of genetic homogenization may be going on in planted populations. We decided to explore this pattern further. We ran an ANOVA followed by a post-hoc test to identify the specific group differences. And as you can see in this table, levels of co-ancestry between nurseries and between planted populations, represented as the first term in the comparison, were consistently higher than levels of co-ancestry between or among age cohorts in natural colonization sites. This also supports this signal of genetic homogenization in planted populations that we previously detected. To explore for patterns of differential selection and potential enrichment of particular alleles in planted populations versus natural colonizers, we tested for significant associations between genotypes and afforestation strategies. As you can see in this figure, with the results for both species, a single explanatory variable, planted or not, clearly identified and separated the two groups of individuals. The explained variance in this analysis, this redundancy analysis, by the way, was low, but significant. It was always, in all cases, under 1%, but it was significant. we identified 224 RD8 outliers that were associated to up to 24 genes of known function in oak. Functions that we identified as a candidate for selection along afforestation pipelines included synthesis of carotenoids and waxes, resistance to disease and pathogens, iron storage, response to stress, and also flowering and fruit development. In the case of birch, we identified 141 RD8 outliers, but we couldn't find any associated gene of known function. These results suggest that some degree of selection, some selective processes may be happening in afforestation pipelines, resulting in allele frequency shifts in genes, in functional genes that can impact the fitness of the planted populations and also their resilience to climate change and other stressors. So in conclusion, our results reveal that diversity is generally maintained in planted populations of pedunculate oak and silver birch in the United Kingdom, yet there is some evidence of potential risks for maladapted planted populations, which can result in more vulnerable populations to stressors like climate change, but also pests and diseases that are very prevalent these days. All of this underscores the importance of considering population genetic background and adaptive diversity when conducting afforestation efforts. And that's all. I would like to thank the co-authors of this presentation, the remaining collaborators of these projects, and other people and companies that have participated in the project. Thank you. Hello.
So
sure.
You had that outrageously high heterozygosity for one of the specimens, or one of the, and you mentioned that it may be foreign transplant.
It's actually this one, you mean?
Yes.
So it's private allelic richness, not heterozygosity.
Okay.
That's of course there, yeah.
All right. So is that likely just the same species from a different location, and there's
Yes.
that much of osteography, not a separate subspecies where it got some hybrid?
Wow, that could be. Actually, the values are, it looks high because most of the values are actually quite low. So the values in a wider frame, maybe they are not that high. So I wouldn't say there's a different subspecies. I bet that they are the same species from a different country, possibly France, which is what sometimes happens in the UK. Thanks. Okay, thank you.
Okay, so thanks a lot for your talk, Guillermo. Next up, we have Katharine Prata, who is currently a postdoc in reef restoration and adaptation program in Australia. But today you're presenting work from when you were at University of Queensland? Still at University of Queensland, okay. Yes, and so Katharine is interested in applying population genetics theory to non-model populations for applied conservation. Now you can take it away.
All right. Hi, I'll be talking about work conducted with Eric Crandall and Cynthia Reginos, and that's estimating coral dispersal distances across the world using the isolation by distance model. So coral reefs around the world are imperiled by marine heat waves and increasing sea surface temperatures from climate change. And so these events are happening right now, and this was the 2024 bleaching event on the Great Barrier Reef in March. And so this is a mosaic of images taken from an aerial drone. And this is what it looks like close up. The corals are under heat stress, and they've lost their symbionts, which they need to survive. And in June 2024, in this particular reef, 97% of the coral colonies had died. And so there's great need to measure key traits that will affect the survival and persistence of these incredibly diverse ecosystems following events like this. And so one of these key traits is dispersal. Following a disturbance, short distance dispersers are able to quickly repopulate locally if remaining colonies survive, yet long distance dispersal can allow unaffected populations to replenish the affected populations. In changing climates, short dispersers may harbour adaptive diversity locally, for example, between the lagoon and the reef slope. And long distance dispersal enables
favourable alleles to travel further or populations to shift their ranges. And so regarding coral dispersal, there are two main reproductive modes. Brooders who release their developed larvae, which are able to settle within hours of release, and then spawners who release their gametes into the water column, and this may take days to weeks before their larvae are developed enough to settle. And so corals are very similar to plants, where they have a dispersive early stage and then their adults are fixed in space. But tracking minute larvae in large oceans is impractical, and so we generally rely on using biophysical models and pelagic larval durations to predict how far they travel, or use genetic structure to predict gene flow. Using biophysical models, spawners are predicted to frequently disperse tens of kilometres, with maximum distances around 100 kilometres. However, biophysical models don't tell us about effective dispersal. Do they survive and do they contribute to the next generation? And with genetic structure, we're able to see large breaks in genetic similarity. For example, here is the genetic structure occurring in both a spawner and a brooder. However, genetic structure does not tell us about ecologically relevant dispersal. And so we can use population genetic theory to estimate effective dispersal distances indirectly that are averaged across the last few generations. The isolation by distance model was created by Wright and extended into a lattice by Mallicott. But because individuals mate with other individuals nearby and go on to disperse nearby, genetic drift causes a decay in genetic relatedness across space, and this spatial and genetic correlation can be simulated based on this model, and then we can extract a few important metrics from it. So one is Wright's genetic neighbourhood, which constitutes a circle encompassing the effectively mating individuals, scaled by 2 sigma. And the neighbourhood is defined by this formula, where NS is the number of mating individuals, sigma squared is the dispersal variance, and DE is the effective density. And so sigma is the key parameter to measure dispersal distances, and it can be used to create a dispersal kernel, which is a probability distribution of dispersal. We can extract the neighbourhood size by regressing genetic distance and geographic distance, and then taking the inverse of the slope of that regression, beta. And we can get effective density by estimating effective population size across a group of individuals and dividing it by the area. And so these two key parameters for estimating dispersal variance seem quite straightforward to calculate. However, the most important thing is choosing the correct spatial scales in which to estimate these two parameters. So that's the area for NE and the spatial distance for the regression. And this is because of the relationship between space and time. When you look at a small area, you're looking at recent relationships, and when you look at a large area, you are looking at more historical events. And so the isolation by distance model requires the migration drift equilibrium, and Hardy and Beckmans performed simulations to show that the migration drift equilibrium was met between distances of 10 sigma and 50 sigma. And so what we can do is measure beta at these distances. Similarly, the linkage disequilibrium method for estimating contemporary NE requires random mating within the area. And so estimating NE at the scale of the neighbourhood area is the most accurate, as shown by Neal. But both of these require an estimate of sigma, which is the unknown parameter that we're trying to work out. And so Hardy and Beckmans and Hardy suggest repeatedly estimating beta across a range of different distances until that equilibrium scale is met. And so why not do the same for estimating NE across different areas until we reach the neighbourhood area? And so that's what I plan to do, and estimate coral dispersal distances across these ideal spatial scales using published data. I used the Web of Science to find studies of hard and soft corals, microsatellite datasets or SNP datasets, and which had individual level sampling, so each individual colony had a GPS coordinate, and individual level sampling enables the query of many different spatial scales. from local to large scale, and it reduces bias compared to population level metrics. And so I found 368 potential studies, 122 had adequate markers, but only 27 had individual level sampling. And out of these, I got 24 data sets. So I reformatted the data sets and removed outliers and potential clones, because that's a problem in coral populations. And then looking at the distribution of spatial distances among the individuals, I changed a range of different distances in which to estimate beta. And then I also grouped individuals across a range of different spatial scales to estimate NE. And then using each combination of beta and density, I obtained many different estimates of sigma and then chose the ones that fit these two conditions for equilibrium spatial scales. Once sigma was chosen, I then recreated distributions of the two parameters and then created joint probability distributions of sigma. And so here are the results for the first 12 datasets, which had IBD signals. The distributions of sigma values are across meters, with the median value for each species dataset. So these dispersal distances are surprisingly small and well under what's predicted using biophysical models and what we see with genetic structure. And so local recruitment is likely a dominant process within these examples, and it's also likely that there's local adaptation to microenvironments as the restricted dispersal can accelerate adaptation processes. The one example from a SNP dataset from the Kimberley region in northwestern Australia has seascape barriers that restrict gene flow across 20 kilometres, but the dispersal distances are restricted across hundreds of metres. And this spawning microsatellite estimate is from the Great Barrier Reef, which is known, as thought and is generally known to exhibit high connectivity. However, this species harbours cryptic species which are undetectable from microsatellites, so this estimate is likely biased. And so we don't have enough estimates to make generalisations about spawners, but looking at the brooders, it's shown they consistently harbour localised dispersal across different species and different regions. And I have a recent paper on that QR code showing this in the genus Agaricea. And in the mixed reproductive mode group, you can see in some regions that they likely brood, and some they may spawn more often. And then these are the neighbourhood sizes for each dataset, which are all quite low, so between 18 and 500. And so if these populations are or become isolated with these lower neighbourhoods, it could lead to inbreeding. So estimates of dispersal distances and neighbourhoods can be used to guide restoration efforts, such as preventing genetic bottlenecking by outplanting numbers of individuals equivalent to the genetic neighbourhoods or greater, increasing the effectiveness by choosing reefs to restore that are within dispersal ranges to other reefs, and assisted gene flow, so sourcing individuals from reefs not within the dispersal ranges of the target reefs to outplant. So to conclude, dispersal distances in corals can be really localised. And yeah, we're not really sure what it is in spawners. It's likely a lot greater. And so we need more estimates from genomic data, specifically for spawners, and to verify these estimates by modelling isolation by distance across realistic seascapes, for example, using SLIM, that would be really great. And then using IVD, we can get estimates of ecologically relevant dispersal, which is really important for the recovery and adaptation in the next few decades. Thank you for your time, and to my collaborators and funders.
So it's interesting that in the studies where you had both SNP and microsat data, that the SNP dispersal data distances were so much higher than the microsats. What do you think is going on there? SNPs are higher than the microsatellites. Yeah, I mean, so the top one with the microsatellite, that's a deep sea coral. It's known to have really short dispersal there. I guess the SNP data will just give you higher resolution, and we do have cryptic species problems in corals that microsatellites are not really able to resolve. So that could result in, I guess, shorter, smaller estimates. Yep.
And we only had 27 data sets
Yeah.
for corals, so I don't know if you would be able to find these trends, but did you notice any differences based on the number of individuals that were included in these different studies? Sense, like, did you see different trends if there was more individuals versus less individuals?
No, not really. Most of these data sets have quite a few numbers of individuals. I mean, one's on the Great Barrier Reef, across the whole Great Barrier Reef. So some of these data sets do have a lot of individuals, but yeah, for ones that didn't have many individuals, I didn't find IBD signals, so yeah. Yep.
Yeah, that was really a great talk. I have a question about, so you split them into like broadcast spawners and readers, but I'm curious if there's anything interesting that might be thought of for like gonochloric broadcasters.
Right, yeah. I'll have a look into that. Not sure off the top of my head with these ones, but yeah.
I have a question on, great.
Thanks.
I have a question on how it seems that for some species, measuring density of individuals is easier than others. Is there difference in the methodology on how you estimate densities of different kinds of corals?
Yeah, so I'm using NE, so effective population size, so I just need, at least 20 individuals or so per population to estimate
Have you tried
NE.
to work with a cover, coral cover as estimate of density?
For census? Yeah, I do have some census estimates, but I've just focused on the effective, but census is so difficult to estimate for corals. Yeah, very few studies really do it.
Okay, well, thank you so much, Katharine. And our final speaker for this morning's session is Michael Connelly, who is currently a postdoc at the National Human Genome Research Institute at the NIH. Michael's generally interested in cnidarian biodiversity, genomics, and host microbe interactions. Happy to have you here today.
Thank you.
Go ahead.
Great. Thank you, everyone. Thank you, Katharine, for that really interesting talk. I'm sure to follow up with you later. I'm sure you have a lot to add to what I'm doing. But I'm really excited to be here to talk about my project using genome skimming or low coverage whole genome sequencing to assess species diversity and population differentiation in East-Pacific Passalopora corals. But I always like to start with the big picture, and that's remind why corals are such important marine invertebrates to study and understand. And put simply, this is because corals build the reefs that support the world's most biodiverse marine ecosystems, sustain productive food fisheries, and protect shorelines from waves and storms, and drive tourism economies for coastal communities worldwide. And I'm sure we're all familiar with the fact that corals are in a global crisis as climate change is causing the ocean to become hotter, more acidic and less oxygenated, and local stressors such as nutrient pollution can also diminish coral health. Corals respond to these many stressors by bleaching, which is essentially a breakdown in their microbial symbiosis, and it increases their susceptibility to diseases that destroy entire reef ecosystems. So when corals die, the physical structure of the reef starts to degrade, leading to the loss of habitat for reef fishes and invertebrates, and threatens the loss of reef ecosystem services for the human communities that depend on them. So to better understand coral ecology, evolution, and symbiosis in the face of this climate crisis, and to devise new conservation solutions, we turn to corals in the genus Passalopora as a model for a few key reasons. The first is that Passalopora corals have a really broad geographic distribution from the western Indian Ocean to the eastern tropical Pacific, and they are hardy, resilient corals that can survive in these marginal habitats. And second, because of this resiliency, Pocillopora corals tolerate captivity better than other species and are easy to culture in aquariums. So we can create small replicate fragments for use in manipulative experiments that seek to better understand coral cell biology and microbiology. However, a major problem with this is that the Pocillopora coral taxonomy is still unresolved and species identification is notoriously difficult. So this story begins when early taxonomists described over 50 species of Pocillopora corals based on morphological traits like branch patterns and polyp arrangements. And the problem that emerged is that Pocillopora corals demonstrate extreme morphological plasticity that makes visual species identification, especially in the field, extremely challenging. And since the same species growing across different environments can present multiple morphologies, in the late 1970s, many of these original species names were grouped together as ecomorphs of only 21 currently accepted species. Yet in the past few decades, molecular genetic studies have uncovered substantial genetic diversity despite this high morphological plasticity, indicating that our current taxonomy doesn't adequately account for the number of genetically and potentially ecologically distinct Pocillopora species. And so the research question I'm interested throughout this work is how can we better identify these ecologically distinct Pocillopora species? And so to do this, I've been working to apply process-based species delimitation approaches that better resolve these evolutionarily discrete coral species lineages. And in doing this, we're following the unified species concept that defines species as separately evolving metapopulation lineages, which basically means that different species follow separate evolutionary trajectories. And in this framework, many lines of evidence can be used to support species delimitation, and different criteria for speciation, such as complete reproductive isolation and reciprocal monophyly, can emerge at different time points during divergence. The good news in the case of Pocillopora corals is that within just the last few years, multiple reference genome assemblies have become available, and their completeness and contiguity continue to rapidly improve. And you can see this reflected in the increasing sequence contiguity and conserved synteny and gene order between these reference genome scaffolds assembled from different species originally collected in Saudi Arabia and French Polynesia and published in papers only 3 years apart. And in addition, the wealth of Pocillopora genomic data is growing exponentially, and recent sequencing of ultra-conserved elements, or UCE loci, from corals across the Central Pacific and Indian Oceans enabled this process-based species delimitation framework to accurately identify genomic species hypotheses that are now being used to rectify these taxonomic problems. A key gap in this effort is the tropical Eastern Pacific region, which is geographically isolated from the world's most biodiverse reef ecosystems and has suboptimal conditions for reef growth and development, such as seasonal cold water upwelling and periodic El Nino warming events, which are oceanographic conditions that emerged relatively recently in geologic time following the closure of the Isthmus of Panama 3 million years ago. And as a result of these harsh environmental conditions, Pocillopora corals have become the dominant reef framework builders throughout the region and are ecologically critical keystone species that often grow as dense thickets of naturally occurring clones that cover shallow rocky areas along the coast. So for this research, it was done in collaboration with a network of scientists at the Smithsonian's Tropical Research Institute and National Museum of Natural History and research universities in Central and South America. And so coral tissue and DNA samples were assembled at the museum from sites in the Gulf of Panama and Gulf of Chiriquí in Panama, Isla del Coco, Golfo Dulce and Bahia Culebra in Costa Rica, Isla Gorgona in Colombia, and the Galapagos Islands in Ecuador, and Clipperton Atoll offshore of Mexico, for a total of 342 samples that were processed for genome skimming across 3 sequencing lanes this past year. So DNA was extracted from the samples and libraries were prepared using an NEB kit, and samples were sequenced to a target depth of around 10 million 150 base pair paired end reads per sample. And so I processed the reads from sequencing and then ran several downstream analyses to assemble coral mitochondrial genomes and extract a key species barcode, which I won't be discussing in detail today, and also map reads to Pocillopora reference genome and call genome-wide SNPs for use in population genetics analyses, And lastly, assemble contigs using spades to extract these UCE loci for multiple sequence alignment and phylogenetic tree construction. So the first thing I had to do was to correct for the presence of naturally occurring clones in the data set, which would otherwise bias estimates of population genetic diversity. And to do this, I used ANGSD to identify genome-wide SNPs and perform identity by state analysis, which calculates a pairwise matrix for hierarchical clustering and clonal genotype identification based on a threshold cutoff determined by including technical replicates. And basically, this analysis revealed the existence of 229 unique genotypes in the data set, with 46 clonal genotypes and 183 singleton genotypes. And this allowed us to correct for samples from the same clonal genotype and retain only one sample from each clone group. So I then used ANGSD on this clone-corrected data set to identify over 4 million genome-wide SNPs, and then retained about 1.3 million SNPs after pruning for linkage to equilibrium. And PCA of this data set revealed 5 genomic clusters that closely matched the nominal species of Passalopora effusa, Meandrina, grandis, and varicosa, and also corresponded to the recently established genomic species hypotheses for these as well. And interestingly, admixture analysis detected 2 population clusters within Passalopora grandis that I'll describe in more detail in just a second, as well as some admixed individuals at Isla del Coco. So maximum likelihood phylogenetic analysis of extracted UCE loci supports these species boundaries and suggests strong population structure across the East Pacific barrier, as East Pacific samples from each species formed a monophyletic clade within its genomic species hypotheses that included the Central Pacific and Indian Ocean samples. Also, interestingly, there were some biogeographic patterns that emerged when we examined which species were present at which sampling locations. The points on this map represent the relative abundance of Passalopora species at different sampling locations and the total number of unique genotypes present at each location. So you'll notice that Passalopora varicosa A in turquoise is present at the continental sites, but is less abundant offshore, and effusa and Meandrina are only present at the offshore islands and do not appear at continental locations. However, Passalopora grandis in orange and brown is the most dominant and widespread species found at all sampling locations, and there is a different cluster. The different population clusters are separated among the offshore islands of Isla del Coco, the Galapagos and Clipperton, and the continental sites in Panama, Costa Rica, and Colombia, despite occurring in symmetry at some offshore sites. So when we look more closely at this variation in offshore and continental Passalopora grandis lineages, we observe low genetic differentiation in the continental lineage despite very high morphological plasticity in our field photographs. And when we delve a little further and identify what loci contribute to this variation along the principal component 1 between these offshore and continental lineages and look at it in a Manhattan plot, we can see that these SNP loci are located in heat shock protein 14A, as well as matrillin-2 and cell cycle division control protein 2, which are both involved in extracellular matrix organization and restructuring and cell morphogenesis pathways. And so many of these loci under selection between these two lineages are enriched in genes broadly involved in metabolism and stress responses, cell morphogenesis, and environmental sensing, which is beginning to provide us early insights into how these corals may be adapting to their local environments in the offshore and continental sites. So moving forward, I want to perform more outlier scans to detect these loci under selection between these different Passalopora grandis populations, and then estimate rates of gene flow and effective migration among these populations, as well as potentially isolation by distance patterns. And I want to reconstruct the population demographic histories of these corals to identify potential impacts of historical El Nino events, and then investigate broader links between environmental and coral genetic variation. And I want to explore patterns of introgression and hybridization, especially in the admixed individuals at Isla del Coco. And then the last thing I want to do is explore this low coverage data for any microbial sequence that may be present from either the coral's algal or bacterial symbionts to see if there's additional variation that could contribute to these, that is present in these different lineages. And so in conclusion, I want to highlight that genome skimming, these low coverage whole genome approaches, are efficient and cost-effective methods for species delimitation and population genomics, and that these process-based species delimitation approaches should guide a future taxonomic revision of Postlepora that is in many ways already underway. And coral reef conservation management strategies should aim to integrate these types of genomic approaches to better preserve reef biodiversity. And so with that, I'd like to thank everyone who's contributed to this broad project and take any questions. Yes.
That was so awesome. I'm like totally amazed. The question I had was about the symbionts. Do you know if the continental versus the one with the symbiotics?
I've begun to explore this a little bit. No information on the density of the cells relative to the host. There is some variation in the symbiont genera, but I have not been able to partition that to different populations or locations yet. I'm still working how to best quantify the mapping rates to the different symbiont genomes from this low coverage data. Yeah.
Great talk. When you're looking at your outlier loci, have you looked at direct environmental associations between those loci and the different environments for each of the populations through like latent matrix models or anything like that?
I've not done any environmental associations, partly because I'm a little overwhelmed by what data to include in those models, whether it's sea surface temperature from satellites. So it's out of my realm of expertise, but definitely an area where I'd like to apply some of these results, sort of like a seascape genomics approach. Yes.
So some of the cryptic species in corals are associated with different light levels, and so I was wondering if you can tell us a little bit about the sampling design in different areas across the...
Yeah, great. That's a great question, and I have a hunch that light and depth do play a role into some of these. A lot of these samples were from collaborators that had different sampling designs and were made available through an opportunity to have them included in the study. So this work isn't particularly well suited to look at like habitat partitioning and depth and light and many of those like fine scale environmental factors. But it is possible, and I would say likely that it has a role. Yeah. Okay. All right. Thank you, everyone.